In this section of our notes, we're going to be talking about kind of the opposite of passive transport. We're going to be looking at active transport or transports that are considered active. Now, this is at the top of page 6 in our notes. And last class, we talked about there are 3 types of transports that are considered passive. And what that means is if a cell is moving molecules by diffusion or osmosis or facilitated diffusion, it's not going to cost a cell any energy. So no energy, no ATP will be used in those 3 processes. Now, if we see active transport is taking place, or endocytosis or exocytosis, then those will cost the cell some energy. So they're considered active. So remember, passive, a passive person doesn't get too excited or too energetic about anything. They're very passive, very calm. Active, think lots of energy. ATP is going to be used by the cell. Now we're going to go ahead and move to the section where we start active transport. This is on page 9. Okay, so as I mentioned, there were 3 types of processes that are considered active. Now, just one of those 3 is just called itself active transport. When you see active transport, it might look a lot like facilitated diffusion because you're going to see molecules are moving and they're going through a transport protein. So let's indicate that. But you're going to see 2 main differences. One, you're going to see that the molecules are being moved from an area where they're in low concentration to where there's already a bunch of them. And usually molecules, when left on their own, molecules won't move like this. Remember, they always will move from an area of high concentration to an area of low concentration. Think of spraying perfume. So initially you're going to have a lot of perfume just gathered in one area, but eventually it's going to spread out and it's going to move all throughout that room, okay? But in active transport, we're trying to move molecules the opposite direction they would go if they were allowed to cross. So we're moving it from low concentrations through high concentrations, and that's going to cost the cell some ATP, okay? Now, remember with passive transports, diffusion, osmosis, and facilitated diffusion, we are moving molecules from high to low. In this case, with active transport, we're moving molecules from low to high, and we can say we're moving them up the concentration gradient, or we say we're moving it against. In passive transport, we are moving down and we are moving with. So if you think of this as being like a hill to climb, if you're going up the hill, you're not going with the hill, you're going against the hill, okay? All right, so we are moving molecules from an area of low to an area of high concentration through that transport protein, and that's active transport. And again, we can also say that we're moving molecules up or against the concentration gradient. This will cost the cell ATP. So notice on all of these diagrams right here, you're going to see cost ATP, cost the cell ATP. And again, we're moving it from low concentrations to high. And then over here is actually just showing you one protein just over time. So here is a transport protein. And when it's open like this, we have 3 sodiums go in. Now notice there's already a bunch of sodium out here. There's a high concentration of sodium. So we're moving them from an area where there's not as many sodium molecules or ions concentrated. So they load up, and the reason why they're able to cross and go this way is because ATP hooks onto that transport protein, and it causes it to change its shape, and now it's open, and those molecules are moving out to this area where they're in high concentration. At the same time that it's open here, these molecules, these potassium ions, can enter. So here's a situation where one transport protein is actually moving 2 different substances. This is called co-transport, by the way, when one protein can move 2 different substances. And if you look inside, it looks like we have a high concentration of those molecules, those potassium. Let's indicate high K plus, low K plus on this side. And so since we're moving them from low to high concentrations, that would take the cell some energy or cost some energy. And it did. This ATP molecule can be broken down and can change the shape of this protein and transport those molecules against the gradient. Now, the other 2 that we talked about that are considered active processes are endocytosis and exocytosis. And you guys have actually seen this before in some of the animations that I've shown you. We've talked about how molecules can be moved to the membrane in vesicles, and they get moved along the cytoskeleton. I'm going to back up a little bit here, because that flashing that it's showing you there that's ATP molecules bind into those motor proteins, and then that vesicle can fuse with the membrane. And endocytosis is an active process as well. It's going to cost the cell some ATP because it has to move the membrane around and trap these particles in this vesicle. And it's not really showing you with this diagram, but how the cell membrane is moved around is that there are all kinds of cytoskeleton attached to it that can pull the membrane in and make that little depression there. So that's going to take energy to do all of that. This is showing you, here's those little motor proteins, and they use ATP binds to it, makes them change their shape. So this is the vesicle, here's the motor protein, here's the cytoskeleton. When this gets to the membrane, then this phospholipid vesicle is just going to become part of the cell membrane's phospholipid bilayer. And so it costs energy to get those vesicles to the membrane for exocytosis, and also from the membrane if endocytosis is occurring. Let me hit pause on this, but it's not letting me. All right. Now, if we are having to trap particles like this, these particles aren't able to move into the membrane. They're too big to squeeze through the phospholipids, and they're too big to even get through a transport protein. So endocytosis and exocytosis is how the cell can move really large particles. So again, moving something big in is endocytosis, and using vesicles to transport molecules to the membrane and out would be called exocytosis. So endo versus exo. Now, some molecules or substances that would be moved. So remember, we talked about how our white blood cells are able to engulf bacteria and destroy it. That's an entire cell. It's a unicellular organism. Or other big living cells being brought in could be like yeast for destruction. We have viruses can get into our cell by using endocytosis. So this would be a bad scenario for us because this cell probably isn't going to destroy the virus. This cell is getting infected by the virus. And so we can see that virus attaches onto the membrane. The membrane will sink in, and now that virus is in a vesicle. And eventually it releases its DNA or its RNA into our cells. And the cell is now going to start to make virus proteins instead of our proteins. Now, on some of these diagrams, you have to be careful because you can't really tell sometimes whether the molecules, if you go this way, it looks like molecules are coming in. If you go this way, it looks like molecules are being sent out. Now, we don't bring molecules in and send them to the Golgi. And by the way, you can tell that this must be the outside of the cell. If here's the Golgi, so this must be the cytoplasm. Whenever you guys are drawing your diagrams, it's really helpful if you'll add arrows, because sometimes I can't tell if it's endocytosis or exocytosis unless you have those arrows, because I might think this happens first and now they're trapped and now they're being brought in and fusing with another membrane another organelle of the endomembrane system. Okay, so it looks like here we are being brought in again. If this is cytoplasm, and I know this is endocytosis because I know that cell membrane gets pulled down and kind of makes a pocket. And now those big substances are trapped in that vesicle. So this would be endocytosis. Okay, this one would have been exo. It looks like this one is moving again. See that? I like this diagram because it has the vesicles. The vesicles are made of phospholipids. And those phospholipids just now become cell membrane phospholipids. So this is exo, and here we have an endocytosis with the bacteria, or if the yeast are being brought in and destroyed, okay? So some large substances that could be coming in, viruses, okay? Our white cells can take them and destroy them, but some viruses gain access to very specific types of cells through endocytosis, bringing in bacteria. Or if you're a unicellular organism, you might be eating. So you're bringing in another cell that is your food, like an amoeba trapping and bringing in a paramecium. Large substances that are usually moved out. So we talked about this is how proteins get out. So remember, proteins are big. They're made of hundreds of amino acids. So this one molecule right here is actually made of 100 smaller ones. Let's put my arrow the other way. So I'm just trying to show you that in there, that would be a protein. That protein is being shipped to the membrane and secreted. And then down here, we are seeing what we talked about in the last unit. The Golgi has tagged proteins. So that's a protein, and then this is a protein that's embedded in the membrane, because that protein must have a non-polar section of amino acids there. And so some of those proteins were embedded in the membrane. Some proteins are secreted. And we talked about some of those proteins that are being released are things like insulin and adrenaline and mucin. Okay, so we're moving things out, exocytosis. And I wanted to show you endocytosis because this shows a little bit more about why it takes energy. So I already included one down here, is that the cell has to wrap its membrane around to bring something in, or it has to pull it down. And so this is cytoskeleton, and those are little motor proteins. And the cytoskeleton is connected to proteins in the membrane. And so the motor proteins can pull using the cytoskeleton that's attached to those phospholipids and make this depression and bring something in. So this is the reason why endocytosis and exocytosis are active processes, is because to manipulate the membrane, to trap, that's going to take some ATP. And to send molecules to the membrane, it's going to take these little transport proteins walking on cytoskeleton. So I'm just going to draw some cytoskeleton here, and I'm just going to put some feet there, some legs for that motor protein. And let's indicate it is using ATP to get that vesicle to the membrane. Okay. All right. And so again, that means that if they're active processes, they are using ATP to move molecules through endo and exocytosis. All right, this last page right here just summarizes. There was 6 that we talked about, okay? So we talked about that in passive processes, no energy is going to be used by the cell. In each diffusion, osmosis, and facilitated diffusion, we're moving molecules from high to low, high to low, high to low. If you are a small uncharged molecule like oxygen and CO2, or if you can get through those nonpolar tails. Nonpolar will let nonpolar molecules cross. Remember, a teacher will let a teacher into the building, okay? Water's polar, so it can't go through those phospholipids. It's going to have to go through a transport protein. If you remember, that's called an aquaporin. Moving from high to low concentrations, that's osmosis. Any molecules that have a charge on them, so amino acids and glucose, they're polar molecules. They have partial charges. So if you look at one side of a glucose molecule, it'll have a partial positive in one area. And then on the other side, it'll have a partial negative. Same with amino acids. So since they have a partial charge, they cannot get through those uncharged tails. And then ions have full charges. So remember, if you're charged up, you're not coming in the building. You can't easily cross into our building. You're going to have to go through a transport protein. Okay. And then down here, we talked about 3 processes that are considered active. Active transport, endo and exo, does cost the cell some ATP. And one thing I want you to note, notice that in these boxes, everything I wrote here is the exact copy of what's down here. So facilitated diffusion and active transport are the exact same thing. We're moving the exact same molecules through a transport protein. But if we need to highly concentrate them, if we need to move them from low to high concentrations through a transport protein, and it costs us some ATP, that's not facilitated diffusion. That's active transport. Endo, again, moving molecules in by using vesicles, trapping them in vesicles. Endocytosis is moving those big molecules or cells or viruses in. And exocytosis is when we have substances like proteins being sent by the Golgi. We talked about proteins being sent to the Golgi by the membrane. We are moving molecules out. Okay. And that completes our notes on active transport.